So I will speak. We haven't cured HIV yet. So until we cure HIV, I have to keep talking about the obstacles to curing HIV. And I'm going to restrict my comments to topics that are less popular in the field. And my talk will be based on my religion. You will hear other religions at this symposium. And Mattia Peterlin will speak later about latency and how it poses a challenge to eradication of HIV. So I'm going to talk about mechanisms of viral persistence. I'm just going to mention latent infection because Mattia is going to articulate in a much more expanded fashion on that topic. But I'm going to focus on the concept of ongoing or residual viral replication and how that might play into our attempts to eliminate HIV from infected individuals. I'm going to talk about comparison of antiretroviral intensification studies and how that can inform us as to the activity of the virus in individuals who don't appear to have much viral activity. We're going to talk about viral persistence and how it may be a consequence of suboptimal antiretroviral penetration into lymphoid tissue. And then finally, I'm going to finish up with a discussion of other potential reservoirs that may persist in individuals on suppressive therapy. This is a phenomenon that is well known to any clinician or researcher who takes care of patients who decide to interrupt therapy. So, in these individuals, these have been individuals who have been on antiretroviral therapy. The virus is controlled. It's undetectable in blood. When any individual interrupts therapy for whatever reason, and it doesn't matter how long they have been on therapy. So when these individuals interrupt therapy, you see this rapid recrudescence in plasma viremia. And that doesn't matter how long they have been on antiretroviral therapy. So that means that there is a reservoir, a compartment in which the virus is residing that appears to have an extreme longevity. But one can illustrate that longevity of these reservoirs that are persisting in therapy if we look at the recently publicized and so-called Boston patients. So Timothy Henrich, along with Dan Kuritzkis in Boston, studied two individuals who were HIV positive but who developed chronic myelogenous leukemia. And in the United States and most other places, the standard course of treatment for this is a bone marrow transplant. Now, you're all familiar with the Berlin patient, Timothy Brown, who received a bone marrow transplant, but his physicians were very smart. Instead of using bone marrow from a matched donor who had normal levels of CCR5, they used bone marrow from an individual who had a homozygous deletion in the CCR5 gene. And the CCR5 gene is an essential co-receptor for HIV entry into cells. And you all know the outcome. Following that bone marrow transplant, Timothy Brown became HIV negative and has stayed that way. Now, the difference between Timothy Brown and the Boston patients is that the Boston patients were transplanted with bone marrow from individuals who had a heterozygous deletion in CCR5. But at least at the time of this publication, everything looked really good. So this is patient A, for 4.3 years, following that stem cell transplant, the individuals remained negative for viral DNA. Now, they're on therapy during this time, and they could not detect viral DNA using very sensitive methods to pick up proviral DNA. They tried various ways to culture virus to see if there was any biologically active virus in these individuals. And at least down to these extreme levels of sensitivity, no detectable replication-competent virus in these individuals. And there was at least a 3-log reduction in viral DNA, complete chimerism, so the recipient cells were completely replaced by the donor cells, complete chimerism. And the same with patient B. For over 2.6 years following the transplant, no viral DNA, unable to culture virus, and in this individual, they did rectal biopsy to see if there was any viral DNA hiding in lymphoid tissue, gut-associated lymphoid tissue. Same story. These individuals appear to have been cured. Now, of course, the assays, the tests that we use to detect HIV are not perfect. There are sampling issues, there are sensitivity issues, and it's possible that the virus is hiding in a compartment that isn't being measured or accessed by these types of tests. So that really, the only way right now to prove a cure or otherwise is to interrupt therapy, to take the antivirals away and look for where the virus rebounds. And as was presented at CROI 2014 in Boston a couple of weeks ago, patient A rebounded after 80-plus days following treatment interruption, and patient B rebounded 220 days after treatment interruption. So this is very different to what we see in individuals who have been on long-term therapy and who interrupt therapy. Normally, you see viral recrudescence in several weeks. So here we have two individuals, one rebounding after 80 days, one after 220 days. It illustrates the extreme longevity of the reservoirs in which the virus is hiding in these individuals who are on long-term therapy and in these individuals who received a stem cell transplant. So we have 45 million infections worldwide. As yet, we only have one cure, and that's Timothy Brown. So how is the virus able to persist in these individuals in the face of overwhelming antiretroviral pressure? And this is where religion comes into play. So reservoir researchers who work on cure basically follow different religions. And like any religion, there is always going to be conflict between those different religions. Now, I was traumatized being raised a Catholic, and that's why I like to use the religion analogy. And for those of you that know me, many of my sort of personality quirks is based upon the fact that I went to school and was abused by nuns. You don't even want to know the details. So the first religion, which is the most popular religion, is that we have a reservoir of latently infected cells, and that reservoir provides extreme longevity to the virus in its ability to survive in the face of antiretroviral therapy. Beware of latency. And some of the experts who have been pioneering these theories are here in this very room today. So one of the models proposes that the cells that the virus resides within, the CD4 T cell, the memory CD4 T cell, has extreme longevity. and that confers upon the virus that infects that cell intrinsic stability. So the virus is basically there for the life of the cell, and as these memory CD4 cells can live for decades, then the virus would be predicted to live for decades as well in this reservoir. And then Rafik Sekli and colleagues have proposed that in addition to this intrinsic stability, there is a continual replenishment or topping up of the reservoir through homeostatic proliferation. Because if a cell harbors a provirus, and remember that the viral DNA becomes part of the cellular DNA. When the cellular DNA is duplicated at mitosis, the provirus is going to be duplicated such that both daughter cells have an identical copy of the provirus. And there's plenty of evidence for that in patients who interrupt therapy, where one sees the rapid outgrowth of clonal viral sequences that likely originated from a population of cells carrying clonal outgrowths, clonal duplications of proviruses. And so as cells die, some of them harboring latent virus, those cells are going to be replenished by daughter cells that continue the latency indefinitely. But the less popular religion, and these are not necessarily mutually exclusive hypotheses, but the next religion is that there is residual viral replication in individuals on therapy. And that is, as this illustrates, a far less popular religion. And for many years, it's one that was not taken seriously by the field, because we all know that antiretroviral therapy works. And if there is ongoing replication, one should see some consequences of that, which I'll explain shortly. But right now, the debate is on the relative degrees to which latent infection versus ongoing replication contribute to the stability of the virus. Beyond this, there are predictions based upon each of those religions as to what is ultimately achievable in terms of a cure. So the first religion has commandments. First commandment is that in patients on antiretroviral therapy, antiretroviral therapy stops all viral replication. These are very potent antiretroviral cocktails that we're using. It's impossible for the virus to survive and replicate in the face of this suppression. Therefore, the stability of the reservoir is not dependent on ongoing replication. It's strictly dependent on the intrinsic stability of the infected cell. And a prediction is that if you intensify therapy, if you increase the level of suppression with additional antiretrovirals, it shouldn't make any difference, because all of the cocktails that we're using just now do not attack the virus in its latent state. The only way they can inhibit the virus is if it's in the process of replicating and spreading from cell to cell. And then on the flip side, the other religion would propose that the antiretroviral therapy is, yes, it's very potent, but it doesn't completely shut down the virus. And as a consequence of this residual replication, there is some replenishment of the reservoirs in individuals, even though they're on suppressive therapy. But the prediction of this is that if one was to increase the level of suppression, perhaps using different classes of agents with different pharmacokinetic characteristics or different antiviral targets, then one should be able to perturb something, and hopefully perturb it in a way that we can physically measure it in one of our viral assays. So two studies that got right at the heart of this issue. One study published last year from Hiroyo Hutano, and another study, an older study published in 2010 from Maria Buzon and a group in Barcelona, asked whether treatment intensification changed anything. Did it make any difference to the reservoirs that are persisting in patients on therapy. And it's a simple design involving patients who have been on highly suppressive antiretroviral therapy. These are individuals who are adherent, so they don't have any viral blips. The levels of virus in blood is always below the level of detection for at least a year, and in some cases on an average of 3.5 to 4 years. So very well suppressed individuals. They used an agent called raltegravir, an integrase inhibitor, because it's different to the ones that these individuals had been on in their prior treatment history. Then they took samples from these individuals at very early intervals post-intensification when they added raltegravir. Now, this is where it gets tricky. What sort of assays are you going to use? Everything's going to show up negative because the virus is latent, there's no viral activity. What these groups did is that they used an assay that measures a circular intermediate, dead-end intermediate, in the viral reverse transcription procedure. The virus infects a cell. 2 copies of genomic viral RNA are used to synthesize a linear cDNA. Now, what's supposed to happen is this linear cDNA translocates to the nucleus, where it integrates into chromatin to form the provirus. And of course, some of those genomes, if they integrate in the right place, as Mattia will expound on, will enter a dormant state that we call latency. But some of those linear molecules are captured by enzymes in the cell that are there to repair broken bits of DNA. These are perceived as broken pieces of DNA, and they're circularized. Now, what happens when you add an integrase inhibitor, and this is work done many years ago by my lab and by Daria Hazuda's lab. If you delete integrase, or if you treat cells in the process of infection with an integrase inhibitor, then the frequency of these circular forms increases about an order of magnitude, about 10-fold. And at the same time, the amount of integrated virus decreases, or in the case of an integrase mutant, does not occur at all. So if an individual has active replication, where cells are in the process of getting infected, and they're treated with raltegravir, what should happen is that there should be an increase in these circular forms concomitant with the decrease in integration. And raltegravir provided a reagent with which to probe this question. So here are the results, the most pertinent results of those 2 studies side by side, the Barcelona study and the San Francisco study. The upshot is that something did happen. The frequency of these LTR forms changed. In those that had detectable forms to begin with, about 1/3 of the individuals in the Barcelona study, there was a spike in the frequency of those episomes and in a subsequent decay to baseline. When that result was first broadcast, was first published, it was considered heretical. You can't have cells in the process of being infected if individuals are on suppressive antiretroviral therapy. But in fact, some of the major opponents of that paper reproduced the study. They showed that in suppressed individuals, addition of raltegravir caused this increase, this transient increase, with about the same kinetics as the patients followed in the Barcelona study. The interpretation of those results is that an increase in episomes can only be explained by de novo infection, that there are cells in those individuals who are highly compliant, highly controlled, but they still have some residual viral replication where cells are in the process of being infected. And this is occurring despite the fact that there are protease inhibitors that are meant to block particle infectivity. Any viral particles made in the presence of a protease inhibitor should be non-infectious. And in addition, you have reverse transcription because the to see an increase in episomes, you have to have de novo reverse transcription, because the substrate for the circularization reaction is a linear cDNA molecule. So both of these processes, virus maturation and reverse transcription, were occurring in individuals on highly suppressive antiretroviral therapy. Doesn't make sense, okay? The investigators doing the studies must be smoking something, hallucinating. But we know that antiretroviral therapy works, but there are certain assumptions when we look at individuals and try to interpret those results. So how do we reconcile what we all know about antiretroviral therapy with those sorts of intensification studies, results from those intensification studies? Because it's clear that many people are studying this. If you look at intensification, it actually doesn't have an effect on residual viremia. All individuals on therapy, most individuals on suppressive therapy have some residual viremia. Adding raltegravir doesn't change that. So people argue, well, then the residual viremia isn't the result of ongoing replication. Well, actually, all the studies, including the Hatano and Buzon studies, did not observe a change in residual viremia when there was a change in these episomal forms. There's no obvious impact on viral DNA levels. For those of you who are trying to follow reservoirs using proviral DNA levels, one has to approach those studies with caution, because the vast majority of proviruses in infected individuals are defective. They're not going to be biologically active. They're not going to contribute to active viral replication or latency. They carry inactivating mutations in critical regions of the genome, and they accumulate. And in fact, they're probably duplicated during homeostatic proliferation, and they are likely to be innocuous if they don't express any viral gene products. There's not going to be much selection for them. Selection against their duplication, sorry. No obvious impact on cell-associated virRNA, but more importantly, lack of obvious sequence evolution. One would expect that if there is residual viral replication, there should be the introduction of random mutations through error-borne reverse transcription, and that might even drive the emergence of viral resistance. So the first assumption then with these studies is that, well, we're assuming that antiviral drug concentrations are similar in all compartments harboring the viral reservoirs. But that is not, in fact, a safe assumption. So a recent paper from Courtney Fletcher and colleagues examined whether in individuals on suppressive antiretroviral therapy, the drugs were at the same concentration in the lymphoid tissue, and remember, the lymphoid tissue is where most of the viral replication, most of the viral reservoirs are located. So they looked at the levels in blood versus lymphoid tissue, and what they found is that the individuals were well controlled when they went on suppressive therapy. However, from that event, if they looked at the frequency of these 2 LTR circles, instead of seeing a reduction you would expect because of therapy, you would expect therapy to impact, to reduce all these viral parameters. In lymphoid tissue, there appeared to be less of a control of viral replication, and that these circular forms actually increased in some of these individuals. But more telling, if you looked at the concentrations of the antiretrovirals in the different compartments, what they found is that in particular in lymph node, some of these antivirals were not concentrating to the same extent that they were found in peripheral blood. So this is showing the median difference in lymphoid tissue relative to the concentration in PBMCs. So we can't then assume that people on suppressive antiretroviral therapy, where drug levels are measured in blood, we can't assume that the drug levels in lymphoid tissue are going to be at the same effective antiviral concentrations. And then the second assumption, they're spreading virus infection. The surprise that there is no sequence evolution, no change in viral DNA levels, would be counterintuitive if there is spreading infection? Well, first of all, these are patients on suppressive therapy, so if there is residual replication, it's going to be at a very, very low level, perhaps insufficient to drive the emergence of drug resistance. But when scientists think of spreading infection, they imagine that there's a cell being infected and passing virus onto other cells. Multiple rounds of replication. At least virologist terms, that's what we think of when we say residual replication or spreading infection. There's actually no evidence from any of the studies done to date that there is spreading infection occurring where the virus is being passed from cell to cell. All the studies can say to date, the Buzon and the Hatano studies can say, is that there is a source of infectious virus and that there are cells in the vicinity that are being infected by that source. But it might only be a single round. This might be a chronic source. The cell that is in the vicinity gets infected, but that cell does not pass the virus onto other cells. In virological terms, this is just single cycle infection. But if this is occurring in these individuals on suppressive therapy, one would not expect to see changes in viral DNA levels. One would not expect to see sequence evolution and emergence of drug resistance. So despite suppressive antiretroviral therapy, which is controlling HIV in individuals who are adherent, there is persistent de novo infection. I'm not going to use the word spreading infection because we don't have direct evidence for that, but at least we can say there is de novo single cycle infection that's persisting in lymphoid tissue. That might be a consequence of an inability to sequester antivirals in tissue that may create conditions for de novo infection at those lymphoid tissue sites. And that has implications for how these long-lived reservoirs might be maintained. So beyond the possible clinical implications of what this residual infection may be doing, for example, to drive immune activation, what are the implications for maintenance of the reservoirs? So going back to the religion, this is the latency religion where we have latently infected cells that, if measured at time X, may have this value, and if measured at subsequent intervals, years after that initial time point, basically have a very slow, a very gradual decay characteristic. And the consensus is that this is being driven by a population of latently infected cells. Again, I have no horse in this race. I keep an open mind. And until one theory is disproven versus another, we have to embrace all possibilities. But again, what this is assuming is that we have a reservoir of intrinsically stable memory CD4 T cells and that there is no replenishment of those cells. But if we do have residual replication, and that residual replication is producing virus that is entering the latent pool, we may have an additional mechanism to account for the intrinsic stability or the longevity of that reservoir. We can imagine the reservoir being water in a sink. Now, normally that sink, the water in that sink remains static. The level doesn't change. But what we didn't realize is that the faucet is leaking and there's water dripping into the sink. And in addition, there is a drain where the water is draining from the sink. So the sink might be perceived as being static, but in fact there's a dynamic turnover of the water, there's a dynamic turnover of the reservoir. Okay, so I'm going to switch to the next heretical component of reservoir research to make the case for macrophage in HIV persistence in patients on antiretroviral therapy. This is Ashley Haas. He makes a nice nun. And Ashley is actually a pioneer of the interplay between lentiviruses and their animal hosts. And the role of macrophage in HIV persistence has been controversial and unpopular. And most of the attention on viral reservoirs has focused on latently infected T cells, and rightly so. But also, most of that attention has been driven by the fact that it's actually quite easy to sample the T cell reservoirs because you simply draw blood from an individual and you can measure by endpoint dilution the amount of biologically active virus. Doing the same sorts of experiments with macrophage that are tissue-bound is far more challenging and invasive. But if macrophage are indeed a reservoir, then we may have to have a completely different set of strategies with which to achieve their eradication, to eliminate those reservoirs. Because in the T cell field, people are using agents that activate T cell latency, that remodel chromatin to wake the virus up from its transcriptionally silent state, something that Mattia will touch on. But those sorts of strategies may not work in macrophage that harbor HIV in suppressed, antiretrovirally suppressed individuals. And if you just meander the literature, you'll find that there are many studies in the past looking for other non-CD4 T cell reservoirs. Eosinophils, stem cells, monocytes, astrocytes, microglial cells. And I think it's understandable to accept the skepticism surrounding non-CD4 T cell reservoirs because many of these studies were poorly controlled. There's always a question of the purity of the cell fraction that you're studying. Maybe there's contaminating T cells. So really there haven't been any convincing studies to argue that in individuals on antiretroviral therapy that there may be non-CD4 T cell reservoirs. But what I want to remind you of is that viruses don't do things without reason. Viruses evolve characteristics. They evolve activities that are beneficial to their ability to survive within the host. So in the life cycle of lentiviruses, there is a recently discovered antiviral restriction. It's called SAM-HD. SAM-HD is, at least as far as we can tell from research from a number of groups, is a myeloid-specific antiviral restriction. It's operative only in myeloid cells to counteract viral infection. And this restriction is active pretty much against all retroviruses and lentiviruses. And my lab started working on this restriction many, many years ago by focusing on the cellular protein, the viral protein that is used to counteract SAM-HD. In HIV2 SIV-SM, that protein is an accessory gene product called VPX. And we didn't know about SAM-HD. This was a study that was published a decade and a half ago. At that time, we didn't know about SAM-HD1. What we did know is that for HIV2 or for SIV to infect macrophage, it was absolutely dependent upon a functional VPX protein. And then my lab did some heterokaryon experiments to show that there was a dominant acting restriction that was being neutralized by VPX that allowed infection of macrophage. And those features were totally dispensable in CD4 T cells. But based on in vitro manipulation with macrophage, we tried to infect pig-tailed macaques with SIV variants that lacked a functional VPX. Those variants replicate indistinguishably from wild-type virus in macaque lymphocytes. But those viral mutants are dead in macrophage, in macaque and human macrophage. So we assumed that these viruses may transmit less efficiently because at that time there was some suspicion that maybe myeloid cells were the conduit for the virus across mucosal surfaces, which is not the case. But the phenotype of those viruses was very interesting. Those viruses, when applied mucosally, did not transmit. not because they weren't infecting macrophage. Well, they weren't infecting macrophage, but there was no amplification of the virus at mucosal surfaces. The control experiment was to inoculate those animals intravenously, because there's no phenotype for those VPX minus viruses in macaque lymphocytes. So we assumed that if we introduce intravenously, the viruses should take. And the upshot was that the viruses were extremely debilitated even following intravenous inoculation. There was no amplification of the virus beyond several days post-infection, suggesting that maybe macrophages are playing a critical role in the establishment and amplification of primate lentiviruses. Now we know about SAM-HD, now we know why these viruses have been forced to evolve a VPX protein, because they can't enter myeloid cells without it. So what about HIV-1? And what about individuals on suppressive antiretroviral therapy? Are macrophage a reservoir for HIV in these individuals? And this is a study derived from the DARE Collaboratory, and Lucio Gama is going to talk a little bit more about the DARE Collaboratory. It's a collection of investigators who are studying reservoirs in a collective fashion and looking at strategies for viral eradication by combining forces, combining expertise in a way that's mutually that's synergistic. And what this group has done, Steve Uckel and Joe Wong, is they looked at the distribution of viral DNA and RNA in lymphoid tissue in patients on suppressive therapy. The object of the study was to look at the distribution of viral DNA in different lymphoid subsets, memory, transitional, effector memory, CD4 subsets. So they took tissue, they took blood, and then they took tissue from ileum and from rectum, and then they compartmentalized them into these different CD4 T cell populations and measured the amount of cell-associated viral DNA. But what they found is that the amount of viral DNA in the CD4 compartments was always a lot less than the amount of viral DNA in that entire cell sample before fractionation. So a sizable amount of the viral DNA was in a compartment that was not a CD4 T cell. And in some individuals, if you look here, for example, in some individuals, in ileum and in rectum, the levels of viral DNA approached levels that were found in T cells. They then went on to follow up this study by demonstrating that those non-CD4 T cells were actually macrophage. Now, this is analysis of macrophage populations in lymphoid tissue. It requires lymph node biopsy. It requires pinch biopsy to get at those lymphoid cell populations. And that is, from an interventional standpoint, a challenge for the researcher and for the patient. So what about more accessible anatomic sites? So David Russell from Cornell University is a TB researcher who has a large cohort of individuals who are being studied at the St. Elizabeth Hospital in Malawi. He's interested in TB, so every individual that comes into the hospital, and visits the ID clinic has a bronchiolar lavage. So they've amassed bronchiolar lavages from these individuals, and he came to my lab and picked up some techniques to analyze HIV, and then looked at these individuals, and he looked at them using a technique called fluorescence in situ hybridization. Fluorescence in situ hybridization uses probes that are staggered along the viral transcript. So you can have 20 to 30 probes on GAG and a similar amount on envelope. So you're essentially multiplying the signal generated by each probe. And what they found is that a sizable percentage of the alveolar macrophage in these individuals harbored viral RNA. Now, these are viremic individuals. The story on aviremic individuals is still being developed, but equally interesting. But what about individuals who are on suppressive antiretroviral therapy? So my lab has formed a collaboration with the Veterans Administration Medical Center in Miami to obtain bronchial lavage from viremic and aviremic individuals to ask whether we can find evidence for viral nucleic acids in this alveolar macrophage. And this is just a snapshot from some of those analyses. We now have 50 patients. So at the top here, we have individuals who are viremic, and at the bottom are individuals who are aviremic. And this is the frequency of viral DNA in the CD3 positive population and in the CD3 negative population. And what you'll see is that in this CD3 negative population, some of these individuals who are aviremic harbor viral DNA that is close to, if not exceeding, levels that are found in the CD3 positive population in those individuals. So HIV sequences can be detected in the lung of viremic individuals and aviremic individuals, and at least from the study by David Russell using fluorescence and cytohybridization, some of those infected cells are transcriptionally active. And HIV sequences are detectable in macrophage from lymphoid tissue and lung in individuals on suppressive antiretroviral therapy. So there are some outstanding questions that are being actively addressed. Always the purity of the sorted non-CD4 T cells. That's always going to be a criticism when one is trying to make the case that the virus is residing in a myeloid cell population, that all you're looking at is contamination of T cells. But of course, in some of the individuals we've looked at, the level of viral DNA in the macrophage population exceeds the level in the T cell population, making it accountable to contamination unlikely. Another important question is the route of cell entry. Macrophage are phagocytic. We could be simply looking at the phagocytosis of an infected T cell by a macrophage, and of course macrophage can engulf many T cells. Maybe they have a preferential appetite for infected T cells. So we're doing experiments now to eliminate that possibility, but it looks like once phagocytosed, viral DNA within a T cell has a very short survival, measured in hours. And then, are these cells harboring viral sequences a reservoir? Do they have the capacity to manufacture virus that can fuel viral recrudescence following treatment interruption? That's under investigation. So I'm going to finish by acknowledging one must always, at least in the United States, acknowledge funding sources. If any of you ever get a grant from the American Foundation for AIDS Research, you must acknowledge that source, because the scientific director, Rowena Johnson, will be there in the audience and will kick your ass if you fail to acknowledge AMFAR funded your research. But the same goes for NIH. In most talks in the US, there will always be an NIH representative making sure that you've acknowledged the generous support from the federal government. So I'd like to acknowledge the DARE Collaboratory. As I said, it's a collection of individuals working on reservoir and cure research. I'm part of the DARE Collaboratory, which is headed by Rafiq Sekeli, Mike McCune, and Steve Deeks. Funding from the NIAID and NIMH, both institutes within NIH, and funding from Merck. I'd like to finish there. Thank you.